From Microbe TV, this is Immune, episode number 5, recorded on February 20th, 2018. I'm Vincent Racaniello, and you are listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, how are you?
How is it up in the North Country?
It is a little too warm. I'm supposed to go on a ski trip this weekend, and it's supposed to be 60 degrees. I'm not sure how that's going to work, so we'll see. Maybe it'll be hiking.
They probably make snow, right? So,
they do, but I think once it gets above 40, 45, it's a little tough to do.
Okay, well, good luck with that.
Yeah, thanks.
Also joining us from Worcester, Ohio, Steph Langel.
Hey there. Yeah, it's warmer here as well. It's about 64 degrees and just busy with a lot of assays in the lab. I'm going to be sampling animals tomorrow. I work with pigs, so it's going to be a busy week, but a good one.
Are the pigs big?
Oh, they're big mamas. I have 4 pregnant gilts right now. And what
gilt is what?
So a gilt is she essentially, this is her first parity. So this is the first time that she will be giving birth. Any that thereafter parities, she is referred to as a sow. So they're all very big. I think like probably between 600, 650. And so I sample, I get blood and mammary biopsies. And when they have their babies, I get milk. So you should see me. I'm like a ninja in there with my bright yellow PPE.
Do they get to know who you are?
They do. Yeah. It's funny because their personalities change quite a bit pre versus post pregnancy. They are very friendly to me in the beginning. I'm the one who feeds them. I mean, I do bleed them, which they don't like, but once they have their babies, it's all bets are off. They're very protective. And so it does change a little bit, but I'm pretty good now at kind of maneuvering around them and kind of predicting their.
Do they try and bump into you and knock you over, or they don't do that kind of thing?
I mean, I've had a couple. A couple who have had aggression issues. They ended up in pigs. Savaging is a thing, especially in first parity. And what that means is they will consume their own babies.
Yeah.
And so aggression can run in some herds of gilts, and I've had that issue. But these girls are good, so.
I have this picture of you in there with one pig in each hand, holding them above your head.
I know. I could do that. Take that picture.
It's pretty funny. Well, cool. Yeah. Good luck with that. I just, it's amazing that you work with these huge pigs. Somebody's got to do it, right?
Big girls. Yep. It's funny. I don't know. I think I may be looking forward to futures of mice handling for a postdoc. I don't know. I think I might take a break.
Well, I'll tell you, they're a lot smaller, but they do eat their babies also. We have that trouble all the time. We go in and we expect there to be babies to do something with it. They're gone.
And they try to bite you too.
They
try to bite you. Yeah.
I don't know about milking them. I don't know. I've seen little milking machines some people use, but
yeah, I've seen those. We talked about one recently on a podcast. They modified a milking machine for mice.
Just crazy.
Now, what we're going to do today, we will have a paper. Steph is going to tell us about a very cool paper. We thought we would try first to have Steph explain the science background that you would need to understand the paper. And we thought we would do this instead of doing standalone immunology 101s, because this way they're connected with the paper. And then later on, they could actually stand alone if people wanted. We could break them out as a separate collection of background materials. So first we're going to do that, and then we'll jump into the paper. Sound like a good idea?
Yeah, that sounds great.
So everyone's on board with this. You tell us how you like it, but give us a few episodes. Don't tell us right away you don't like it. Just see how it goes, because we figure we have a paper with certain issues in it that you need some background for. You don't want to wait for an Immunology 101, which could be a year from now. Who knows, right? So this way we get it right up front. Okay, so Steph, it's all yours. And remember, Steph, we're going to interrupt you.
That's fine. That's fine. There's a lot of stuff in here. So yeah, feel free to interrupt me, but I'm going to kind of outlay how the direction I'm going to go. I'm first going to talk about the general mucosal immunology and specifically focusing on the gastrointestinal tract. I'm then going to go on and talk about the regulation of a certain type of T cell called a T regulatory cell. And with that, in the context of how the immune system develops during an allergy infection, what happens when it goes bad and what happens when it goes well, and then how that is implicated in treatment of allergy, and then we'll kind of roll into the paper. So that's the outline of what I'm going to talk about. So first, when I talk mucosal immunology, it's basically the study of the immune system on the surfaces that secrete mucus and things. So this would be your nasal oral passages. This would be the lungs, gastrointestinal tract, reproductive tract, and often forgot mammary glands. And these things, they're thin and permeable, and it's because there's only a single layer of epithelial cells that line the surfaces of these organs. And some people consider skin to be a part of the mucosal immune system because it does connect to the other parts of the mucosal immune system, but it's a little bit different because there are more layers of skin than there are, let's say, to the gut.
There's a couple of interesting things where, at least in human, do you know that like about 99% of pathogens gain access through mucosal surfaces?
Right. That's right.
Yeah.
It's a little different in some of the animals because there's a lot of vectors of vector-borne diseases.
Right.
But in humans, it's mostly through mucosal surfaces. The other thing we don't think about is that we're constantly exposed to innocuous things through these surfaces too.
Right.
We eat, and it's going to be part of what you're going to talk about. All these things that we eat that we shouldn't have immune responses to. It's amazing that happens that way.
Right. And, there's this number, and I think this is one of these big numbers that gets brought up and repeated in papers over and over, but I think it was between 300 and 400 meters squared if you spread out all of the mucosal surfaces. And I looked it up. It's essentially like a basketball court. So if you spread all your mucosal surfaces, it could cover the length of a basketball court. So it's very vast and expansive. And yeah, like you said, especially in our gut, it's coming in contact with both foreign proteins, potential pathogens, also self molecules that are of yourself. And so it's diverse and it's complex. And what adds to the complexity is the fact that we're colonized by a whole microbial population, and they differ quite vastly between sites. So for instance, in your gut, you have more of the phylum Bacteroidetes, but in like the vagina, you have more lactobacillus. So it just depends. It's context dependent. And that also then determines what cell types are at the mucosal lining and how those interact with each other.
And this microbiome at all these mucosal sites, they are separated from the cells by the mucus, right? By that thin mucus layer.
Yeah.
And then when they penetrate, that's a problem.
Yes.
It's a problem for sure. And that mucus, it's produced by goblet cells. There are a whole host of cells that in the lining, and goblet cells produce that mucus, and it helps to trap pathogens, but it also helps to keep the things secreted from the bacteria like metabolites, carbohydrates in close contact with the surface, because all of these things are playing a role in keeping a nice homeostasis so that, things are as they should be. And of course, when they alter is when you have problems. So I'm going to go through and talk about as if you were a food antigen or any type of antigen entering the like if you ingest it, it goes to your intestinal tract, and what happens? What would you see first? So the epithelial cells, they're hugely important in the host defense. They not only provide a physical barrier, but they themselves are producing innate cytokines and molecules that help protect against pathogens. And not only that, you have just the basic peristalsis of the cilia. Cilia are really important. They are in all mucosal surfaces, and they help move food and pathogens and allergens along so that they potentially aren't going to stick and cause problems. So when you're talking about the intestine, there's a really huge dynamic range of from the crypts at the very bottom of the villi to the top. And how those are regenerated is there are stem cells at the bottom in the crypts. And so whenever you damage your intestinal epithelial cells, you're going to then replace those villi at the top with the stem cells at the bottom. And a healthy villi is a long one with a lot of these stem cells at the bottom and really healthy, nice looking villi at the top. There are, like I mentioned before, a lot of immune cells. Innate lymphoid cells are kind of a neat cell type that has been discovered within the last 10 years and shown to be really hugely important in the interactions of cells. And innate lymphoid cells are kind of below the surface. And those intraepithelial lymphocytes are really hugely important in the regulation. And it's bidirectional. So the intestinal epithelial cells are talking, in a sense, to these lymphocytes and intraepithelial lymphocytes below them. So once you get below that, then you can really divide it between inductive and effector sites. And it's really based on the anatomical and functional properties of those different sites. And I'm going to just talk about the intestine because that's what our paper sets up. And that's actually the inductor site that I work with. And so when you have the migration of immune cells from an inductor site to the effector tissues, it's through the lymphatic system. And basically, when you hear the word GALT, it just stands for gut-associated lymphoid tissues. These are the Peyer's patches, the mesenteric lymph nodes, and the isolated lymphoid follicles. So when you have these immune cells in the gut, they are constantly they are sampling the gut contents and they're pulling down antigen. And they're taking this antigen and they're processing it. Things like dendritic cells, specifically, we're going to talk about a certain cell type, CD11C dendritic cells. And really, when I say CD something, it's just representative as what's expressed on the surface of those cells. So you have sampling from the dendritic cells. You can actually have sampling from the intestinal epithelial cells themselves. They process the antigen and they need to present it to something. Now, if you've listened to our past episodes, you're kind of familiar with this concept of antigen presentation. But this happens in the inductive sites, the Peyer's patches, the lymphoid follicles. The dendritic cell is going to present the antigen to a B or T cell, where they're going to mature and then migrate out to the effector sites, which is back to the lamina propria, or essentially the intestine, so they can carry out their effector functions. So it's kind of you're sampling your environment, you're presenting it to the internal compartment of the intestine. So then those cells can go back out to the intestine to help protect you from, let's say, a virus or bacteria.
In the intestine, these cells are below the epithelial layer. How do they sample what's in the lumen of the intestine?
Yeah, well, it's fascinating. There's a couple ways. Dendritic cells themselves can reach their little pseudopodia right through the tight junctions and sample themselves. But it's also been shown that antigen can go through M cells. M cells are specialized immune cells in the epithelial lining that even look different than intestinal epithelial cells. They're a little bit bigger. And antigen can come right through there into a Peyer's patch, which is kind of right below. And the Peyer's patch, again, is a lymphoid follicle where you have dendritic cells. So there's M cells, dendritic cells. I think they have talked about intestinal epithelial cells themselves actually being able to pull antigen in. So multiple ways that these cells can pull it in. Cindy, did you have a?
Yeah, I was going to ask. So when we think about this, there's a couple of things. There's one is, are these dendritic cells performing the same as the dendritic cells we've discussed before? So we typically think of when a dendritic cell takes up an antigen and presents to a T cell, there's an inflammatory response that happens. But we know that that's not always the best thing to happen in a surface where you have other biological processes happening that are critical. For example, in the gut where you have nutritional exchange. And if you damage the epithelium, that's going to damage that potential, right? So the first part is, what do you think the default is for these dendritic cells? Is the function different? And the second thing is, how does the dendritic cell know that it's food or pathogen?
Right. Well, firstly, with these specific dendritic cells, they do have cues from their external environments that helps them be essentially different than dendritic cells of other tissues. So retinoic acid is one such metabolite that actually can, because these dendritic cells, they have the enzymes that can help convert something like vitamin A to retinoic acid. And that really helps to prime that dendritic cell to keep it in the intestine by expression of certain markers, but essentially is helpful in contributing to this homeostatic environment where you have secretion of certain regulatory cytokines. So,
yeah, and I think that the retinoic acid plays a critical role in DC generation of Treg cells, right, in the gut.
Right. So yes, and your second question, if I'm I think it's something we don't fully understand yet, but how do those dendritic cells know that what they're taking up is food versus something that, or something they should respond to versus something they should not respond to? Because we started at the beginning saying that, most pathogens are coming in these mucosal surfaces, right.
Yet these mucosal surfaces are bathed in 1, microbes from the microbiome, and 2, innocuous antigens, and yet 3, when pathogens come in, we need to be able to recognize them.
Right. Well, what's interesting is that a lot of these commensal protein, a lot of the antigens that come from food are also complexed with things like immunoglobulins, which then can dictate how the dendritic cell responds. I mean, it's going to uptake it and process it, but it's going to be received through certain receptors and then processed through MHC2. We've talked about MHC2 before. And so it depends on the molecular patterns of that antigen. And I think you're right. I don't think we know exactly the mechanisms, but it's really an area of hot research right now. So it's fascinating because I think if you look at dendritic cell subsets and any type of cell subsets at different organs, they're going to respond differently and act differently based on what they're sampling.
Cindy, do the DCs make decisions, or they simply take everything and bring it into the lymph node and ask the B and T cells whether it's foreign or not?
Oh, no. I always say people who study T and B cells, and so if they're listening, I feel bad, but I always say the T and B cells are kind of stupid and they don't know what to do. They need the information from the dendritic cells. And the dendritic cell's job is, 1, to take up those antigens, but also to take up the context of those antigens. So if, for example, the antigen comes along with something that triggers a pattern recognition receptor or a TLR, as we've described before, that tells the dendritic cell right away, this came from something bad, and we definitely need to make a response to this. Whereas if, for example, you just inject into a mouse an antigen without anything else, the dendritic cells see that in the context of no danger, no patterns from microbes, and they interpret that as, okay, this is something we should not respond to. And in fact, they generate what was peripheral tolerance, which I think Steph is going to talk about in a couple of minutes. But it basically tells the T cell, any T cell that might see that, shh, don't do anything. So that context is really, really important. And what we don't understand, and I kind of set her up for this, but what we really don't understand is how that happens in the gut. Because when a dendritic cell is sampling, for example, a food antigen, it's also able to see the gut bacteria, components of the microbes in the gut that are released. We have microbes in our food, and there's chunks of those microbes that are available to come along with any antigens. And so how do we make that distinction? Do we make that distinction? And I think it's becoming more clear that it's blurry, because we do make some antibodies and things to innocuous antigens that we might eat. And we'll talk about that in this. There's a sensitization step in the paper that we'll talk about. But the idea there is that you can develop antibodies and immune responses to some things that are innocuous that you shouldn't respond to. And when does that become pathology versus just, for example, antibodies against foods that just help clear them and they don't cause a problem? We really don't understand that. That's sort of the forefront of what people are studying in this area right now.
Right. Yeah. So it's hugely interesting, and I think when looking at how things move around in the gut, we have to take into consideration the receptors that help move them there. And I'm just setting this up because this is a marker. This is a receptor that we're going to mention in the paper. And two markers specifically, polymeric immunoglobulin receptor and neonatal FC receptor. Now, what these two receptors do is they help take immunoglobulins antibodies from one side to the other. So one side of the intestinal epithelial cells to the lumen, for instance. And so when thinking about IgA immunoglobulin is very much a part of the mucosal immune system. It's the isotype that dominates the mucosal secretions. And so this PIGR, polymeric immunoglobulin receptor, it's a mouthful, say that 5 times fast. It sits on the basal side of the epithelial cell, and it binds to monomeric IgA. It then can transport that to the lumen side as secretory IgA by leaving a little J chain, so that this IgA is now dimeric and has double the capacity to bind to potentially pathogenic things in the gut. And what's interesting, not just pathogenic viruses and bacteria, but the homeostasis of that gut lining can be predicted by how many antibodies are binding commensal bacteria. And if you remove some of that can cause potential dysregulation. So the next one is neonatal FC receptor. And what that does is it brings IgG. So we have to have something that brings IgA, and it also, PIGR brings IgM as well. FcRn, or neonatal FC receptor, transports IgG. Now, it's less available in the gut, but you can see it highly in the placenta to help transport IgG from the mother to the neonate through the placenta. And so these are two ways that the immune system is able to help transport these immunoglobulins from one space to the other.
And we know that those antibodies are really important to be transported. One, because coming from the lumen to the underside of the epithelial cells is important, as you mentioned, for delivering antigens. And the other way, delivering the antibodies to the gut helps to coalesce those bacteria together into large clumps and clear them, or at least prevent them from being able to invade. So getting those antibodies back and forth across that barrier are really important for the host defense mechanisms.
Right. So what I was going to move on to then was, we know that secretory IgA antibodies and other different types of molecules are going to help limit the epithelial contact and potential penetration, so that there's no host microorganism invasion or maybe potentially other dangerous antigens. But there are other immunosuppressive mechanisms that help to inhibit the overreaction of innocuous luminal antigens. And an innocuous luminal antigen is just a long term for a piece of food or commensal microorganism. And so what we refer to those is regulatory T cells. And before I move on to really going into Tregs and where they come from and how they develop, I'm curious, did either of you want to do anything more with the general mucosal immune system before we kind of talk more specifically about this cell type?
Nope, I don't think so.
I think it's good.
Okay, great. So regulatory T cells, they are a huge part of keeping the homeostasis in the gut, but they had to get there. So where did they come from? All classes of blood cells are derived from bone marrow. There's resident hematopoietic stem cells, and then they migrate out. B cells are going to be matured and developed in the bone marrow. They're going to stay there. But T cell progenitors, they're going to move from the bone marrow to the thymus. So that's why they're called T cells, because they are matured in the thymus. And so when they get there, they pass through a series of steps. And what it's marked by is changes in the T cell receptor genes. And we've talked about T cell receptor before. I'm going to refer to it as TCR. And it's the expression of this TCR when it gets to the thymus that helps basically characterize what an immature to a mature T cell is. And so when those genes are rearranged, they give rise to two major populations. There's alpha beta T cells and there's gamma delta T cells. In humans, alpha beta T cells are more predominant. What's interesting is in large animals like swine and cows, it's actually gamma delta T cells that can represent up to like 40% of T cells circulating, which I'm pretty sure, if I remember correctly, in humans and mice, it's like 5%. So it's interesting when you think about differences in species. So once those T cells, once they are starting to go into either of these two different lineages, these T cells start to express distinctive markers, and their lineage commitment, or essentially where they're going to go from this point forward, is dependent on the ability of that TCR to bind to a peptide presented to them by the MHC2 complex. MHC2 complex on an antigen-presenting cell like a dendritic cell. And so I remember when I learned this concept, it's kind of like Goldilocks and the Three Bears, where essentially a T cell that's able to bind very strongly to MHC is going to get clonally deleted, because if it is going to respond to a self-peptide really well, it's going to potentially cause problems when it goes into the periphery and it sees self-antigen, and that's what causes autoimmune disease. If it doesn't bind at all, it essentially is ignored and nothing, it doesn't move forward in the maturation process. But if it binds at what would be considered an intermediate level, it then is positively selected to move forward, and then it can go into the periphery. That whole idea of in the thymus, you have a lot of interactions, and the TCR is going to either strongly, not strongly, or intermediately bind to MHC2 presented self-peptide, is called central tolerance. It happens in the thymus. Now, obviously, in the thymus, you're not going to see commensal antigens to food. There's no food antigens likely migrating up to the thymus. And so an antigen presenting cell can't present those type of commensal bacteria or things found in the gut there. So there has to be another mechanism to be able to get those T cells to become tolerant to those different types of antigens. And so once the T cell then comes out of the thymus and migrates into the periphery, it's developed into something called peripheral tolerance. And that's it's different in the sense that you're still going to have interactions with MHC presented peptides, but there's also going to be concomitantly signals from cytokines, things like TGF beta, or transforming growth factor beta, along with kind of this intermediate level of presentation of MHC2 self-peptide or commensal peptide, and that's going to help develop T regulatory cells in the gut. And so it's in the periphery, it's this ability, they're called different things. So in central tolerance, those are thymus-derived Tregs or natural Tregs or TTregs. There's a couple different names, but they refer to the thymus. Once they get out to the periphery, they're referred to as induced Tregs because they're induced by the environment that they're in. And so those are called iTregs or PTregs, P being peripheral. So those are the kind of the two different types. And you don't find a lot of peripheral Tregs, and other spots where you do find them, but they're really highly represented in the intestine. And that's because these PTregs are there because they're trying to create a regulatory environment by the expression of TGF beta and interactions with dendritic cells like those specialized dendritic cells of the gut. And so they work together in this way to help keep a very a healthy, sustainable level of T regulatory cells in the gut. And so I'll pause there and ask you guys how things are going.
Yeah, go ahead.
I'm regulated. I'm fine.
Your Tregs are doing great.
Are there diseases of Tregs? Human diseases where we know the Tregs are screwed up?
Yeah, I mean, in mice, if you take away T regulatory cells, you have autoimmune diseases. In humans, it's called IPEX. And Cindy, do you remember immunosuppressive?
I don't remember what IPEX stands for.
I know it's IPEX.
I know it's, I don't remember what it stands for. But yeah, it's a defect in the generation of T regulatory cells. And so without those T regulatory cells, the T cells that are there do their job too well. There's a really interesting idea in immunology, and that is that everything is very tightly controlled. That Goldilocks idea, right, Steph? So if it's too little, the pathogen wins. If it's too much, there's too much pathology and there's damage. And so you need this zone of response, and it's very fine, and you have to control that. So we want the immune system to be activated, but not too much. And so there's this yin-yang balance there. And it starts immediately upon exposure to microbes. There are regulatory cytokines and regulatory aspects of the immune response that tamper and tune the immune response so that it's appropriate.
Right. And they do directly show, it has been shown that, okay, once we're to the gut, once those Tregs are really proliferating in the gut, when you take certain bacterial strains and add them or take them away, they help to promote the generation of these iTregs or pTregs, and also the things that those commensals secrete. So things like short chain fatty acids, the fermentation of those dietary fibers in the gut are really going to help promote Treg numbers. And it's interesting then once you consider the fact that different people are colonized with different types of bacteria and the effects on then how those Tregs affect regulation in your gut.
And I guess one of the thing that I was going to ask you about is these T regulatory cells. How specific are they? Right. So these are secreting relatively general inhibitory cytokines, for example, like IL-10. Yet there may be a specific aspect to this, right?
Yeah, well, they do. I mean, there are, just like there is in other types of T cells, there are memory Tregs that, basically they have been presented an antigen before, and while they're not actively secreting their effector cytokines at the time, when upon re-exposure, they're going to secrete whatever, maybe TGF beta. So there are effector and memory Tregs, but there's also there's different subtypes that they've been able, and again, T regulatory cells, I mean, when you look in the literature, it really wasn't until the 1990s that we really helped. The two people I want to bring up, Shimon Sakaguchi and Alexander Rudensky, now they haven't won the Nobel yet, but I don't know. I mean, they were the ones who really discovered T regulatory cells, and we don't even understand, I mean, how they function, how they fully develop, and there's different subsets. So one of the defining factors is this master transcription factor, FOXP3. And so FOXP3 transcription factor is really, it helps to, it's the defining factor as to whether CD4 T cell is going to become a T regulatory cell. But there are also now T regulatory cells that are not FOXP3 positive that secrete IL-10. So, and those are THR1. T-Reg.
TR1.
TR1, thank you, Rex.
Yep.
So, and it's every time we think we've defined all the different types of cells, there's some with the technology we have, some other way we can then distinguish them from each other. So, if that
immunologists are very, very good at that. They like to bin things into smaller and smaller and smaller groups with more and more and more markers. And define function of each individual cell type. Yeah, but you're right. You're absolutely right. It's very complex. We think often of just T regulatory cells, but there are many different subtypes, and they have slightly different functions.
I have to say, having lived in the 70s and 80s and before, but when I was in grad school, there was something discovered by Richard Gershon at Yale University, and he called them suppressor T cells.
That's right, and nobody believed it.
Nobody believed it.
Right. Nobody.
And they were forgotten about and then rediscovered, and now they're called Tregs.
That's right.
And unfortunately, he died in the 80s.
I was going to say, didn't he die kind of before it even?
He died in 1983. He was very young.
Right. Okay.
And never got to see the rebirth of what he discovered. Yeah.
I was reading up on him, and I can't remember what gene he was trying to attribute these cells to, but essentially they had tried to name the locus and the gene where these Tregs were regulated from, but then they sequenced the mouse genus and it wasn't there, and it really just fell apart after that. So really, it's unfortunate, because if he had the technology that they had probably in the 90s, he would have been able to. So just, does the luck of your discovery match up with the technology available is a problem in science. So with that, I think I've introduced a lot of the main players. So, thinking broadly, the mucosal immune system, the physical barrier, the mucus layer, then we go below that epithelial layer. You have all these different epithelial lymphocytes. Below that, then you have these inductive sites where the action is happening. Once those antigen presenting cells bring that antigen, they've sampled it, they bring it down present it to the T cells. And those T cells can be T regulatory cells. So I think we've defined a lot of the things that we're going to talk about in this paper. So I'm going to transition over to food allergy and exactly what that means if we're all fine with that.
Sounds great.
Absolutely.
Go for it.
Great. Excellent. So a food allergy, I mean, if you just define it in its basic term, it's a pathological reaction of the immune system triggered by the ingestion of food or a food antigen, particularly. And it can cause a lot of issues. I mean, you can think of people who they're vomiting, maybe they have a stomach issue, but it's also can then be defined once you think of anaphylaxis and hives, and any type of atopic dermatitis. It's been shown a lot of times that when you have a food allergy, it's actually predisposed with sensitization from the skin of that food allergen. I'm going to go a little bit more into that in a second. But it's different from food intolerance. So a food allergy is a pathological reaction, but intolerance, it's not immune dysregulation. It's essentially you can't break something down. It's a non-immune function. So like lactose intolerance, you just don't have lactase to break it down. And from lactose to I think glucose and galactose, that's not food allergy, that's intolerance. And there has been, and I don't know why this is, but they speculate it actually is evidence of increasing amount of food allergies since, I guess, over the last 20 years, or is that really a function of us being more aware of food allergies? But a lot in the papers, they will cite the fact that over the course of like 20 years, you've had 18 to 20% increase in prevalence of food allergy, particularly in children. And so it leads us to asking the questions of why and how can we prevent it, and so on. So I'm going to let you guys choose. Would you like me to talk about when everything goes right first, or would you like me to talk about when everything goes wrong first? It's like choose your own adventure.
Go ahead, Cyn.
Oh, it's me?
Yeah, you can.
Why don't you talk about how it goes right first?
Okay. Great. So in the context of what we've talked about before, you're going to have antigen presenting cells or intestinal epithelial cells or whatever way the antigen gets from the lumen to the below the epithelium. It's then these subset of dendritic cells that are going to migrate to, let's say, the Peyer's patch or mesenteric lymph node, present that to a T cell. And because of the factors in its environment, namely TGF beta, I think IL-2 has been implicated as well, and IL-10, it's going to promote the differentiation of Tregs. And it's then again, these Tregs that help to, when the B cells are going to express antibody for that food allergen, it's more likely to be either IgA or IgG4. And these are just different subtypes. Now, when it goes right in a not in your guts, you're not going to have a response in terms of an IgE mediated response. And so the goal then, when you're thinking about any part, is you're trying to increase Tregs, you're trying to increase IgG4 allergen specific isotypes, and you're trying to decrease things like IgE, basophils, and mast cells. And really this Th2 response. And the Th2 response is really defined as cytokines like IL-4 secretion, and then the degranulation of mast cells and basophils. And that transitions me as to when things go wrong. So when you have IgE-mediated food allergy, you have food allergen-derived epitopes. So a piece of, like a piece of an allergen of a peanut. And when it's then presented to B cells from dendritic cells, that B cell is going to isotype switch to IgE, and that IgE is then going to go bind to the surface of mast cells. Now, what's interesting about allergy is that you can be sensitized to something, but it's upon the second exposure that you have the strong response. And so what happens is, let's say, I had mentioned before that in a lot of food allergies, you actually have sensitization on the skin as to the antigen before you notice that something, you eat a peanut and something goes wrong. They have epidemiological data has showed that young children who use lotions with peanut antigens in them or wheat antigens, they're more likely to have food allergies later on when they eat those. And so what they suggest is that once you see that allergen for the first time, you have IgE antibodies secreted from IgE secreting plasma cells bind to the mast cells. But when you have the second exposure, you have a lot more of those IgEs binding to the mast cells. And what happens is something called cross-linking. It brings together something called Fc epsilon receptor. We talk about Fc receptors on the surface of these cells. Once those Fc epsilon receptors are brought closer together, the cross-linking of the binding of the IgE makes it degranulate. And that's what causes things like anaphylactic shock, because you have vasodilation, you get hives. There's a lot of different things happening in the body that produce this type of reaction. And so,
and the mast cells, when you say degranulate, what they have inside them is little packets. Of stuff, and the stuff within those packets includes histamine.
Yes.
And so histamine is the canonical effector of what we typically think of this immediate hypersensitivity or allergy. So when you're exposed and those mast cells fuse those little pockets with the plasma membrane and spit out all of the contents, that includes histamine. And the histamine binds to the histamine receptors, and that's what causes the itching, the vasodilation, the redness, the sneezing, and all of that. And that's why we take antihistamines to combat allergy.
Right. And histamine is, that's something that I think immediately when I think about allergy. But there are other things that, depending on how quick the reaction is, so that histamine response tends to be quick. It's within, like the first, a couple hours or hour. But then later on, you can have kind of a secondary response. There's things like platelet activating factors, leukotrienes, IL-4, IL-5, which I know when you hear these ILs, it's like a soup of different interleukins is what IL stands for, but it is all contributing to this Th2 dominated response. And a Th2 response isn't always bad. I mean, when we have a helminth infection, a worm infection, those different factors are going to help to clear whatever worm or parasite. But in terms of an allergy that can't be then regulated, that response is when you have, like what we've mentioned before, the itching, the redness, and it can really become bad if you have bronchoconstriction. And you can't breathe, essentially, and a lot of mucus production. Though that combination is not good. So So that is in the context of when things go bad. Now, linking Tregs to this response, it has been shown that when you have allergic response, there is a dysregulation of T regulatory cells, and that there's potentially this interaction that an activation of mast cells through IgE leads to the suppression of Tregs generation and their amplification. So then the Tregs can't help control the Th2 response, because then they're being suppressed themselves by mast cells or degranulation or those Th2 responses. So it's always this feedback as to who's winning, who's more in response, and then that's going to suppress the response that's supposed to control it, and it essentially gets out of control. That really helps to set up what oral tolerance is, or tolerance in general, because the goal then, if you're pre-sensitized to something and you genetically have a disposition to have a stronger response upon the secondary reaction, you would want to then develop some type of tolerance to that allergen, so that you obviously do not respond that way. And it seems like, they've shown that if you orally give somebody immunotherapy, or you give increasing amounts of whatever allergen that is over time consistently, you can expand the Tregs, decrease the Th2, and that you have no response once you're then challenged or you eat that. Or at least if you eat a little bit of it, you're not going to spin out of control. And now whether this oral immunotherapy, feeding people oral challenges to the food they're actually allergic to, is something that's long lasting, I think that's not fully understood, but the goal essentially is just to desensitize you by giving you increasing amounts. And it seems like where you give the allergen really, and this is with all mucosal sites, it really determines then how you're going to respond. So like I mentioned before, if you're sensitized in the skin, it doesn't have the same, your Tregs in your gut are not going to be as expanded as if you're orally sensitized with the allergen. So I think that's why it works when you give somebody increasing doses, because you can slowly build up the proliferation of those Tregs by the presentation from the dendritic cells. So I can go into some of the real life things, meaning diagnostics, prevention. I don't know if we want to get that into that, but it's I don't have allergies. Do you all have you experienced some of these things?
I don't. I'm lucky.
No, I don't either. No.
Wow, I think that's statistically
unusual,
very unlikely. Yeah,
right.
I know when I've asked my classes when we talk about hypersensitivities and allergy, I said, "How many of you have allergies to at least one thing?" And I would say, on average, about 80% of them raise their hand or more. Now, granted, the ones that don't have them maybe aren't as interested in the topic and they skip that class, but it is what it is. But then it gets very interesting because I'll ask them, well, how many are allergic to more than one thing?
Right.
And, half of the hands that were up stay up. I said more than two things.
Right.
And, a third of those still stay up. More than three things, more than four things. And still, even then, there are several people out of 100 that still have their hands up. Wow. And that's the idea of atopy that you were mentioning. So when you have this genetic predisposition to generate that Th2 type of response to these innocuous antigens, you're much more likely, and your body is setting itself up for, upon exposure to new things, you do the exact same thing. And so there's this idea of spreading. You might be allergic to one thing in childhood, but then by the time you're an adult, you have a whole suite of antigens that you're allergic to. You might go get an allergy test where they put a little bit under your skin and your whole arm will blow up because everything you're allergic to.
Right.
And so yeah, it's that idea of that epitope spreading. Now, the other thing we don't understand is you might have allergies in childhood and they're completely gone when you're an adult.
Right. Right.
How that happens, we have no idea. You may never have an allergy until you're 40 years old and all of a sudden you're allergic to ragweed. We don't understand that either. There's so much about how these allergies are developed and how they are regulated and how they appear and disappear that we just simply don't understand.
Right. And when you look at when you read up on the different factors that could affect this, it's vast. I mean, microbiota, vitamin D, vitamin A, the ability of your cells that are changing over time with age, and all of these things maybe, like you said, contribute to this kind of unknown, or why is somebody developing allergy? What was interesting is that there has been shown that your response, that some people are allergic to, I forget what carbohydrate in meat it is, but they are more likely after they've been bitten by a tick who's recently fed on a blood meal and then bites you, and then you are more likely to be allergic to red meat after that. And I find that fascinating. Yeah.
So the worst thing that can happen is you die from an allergic reaction, right? So what's the intervention for that?
Well, anaphylaxis, I mean, you can give epinephrine. I mean, is it epinephrine?
Yes.
Yes.
EpiPen. Like an EpiPen.
EpiPen.
Yeah.
Thank you.
That's right.
You can give that to help decrease the symptoms that lead to anaphylaxis. You can take antihistamines. I think that's a pretty booming industry, around ragweed season, and you take antihistamines. And I think there's other ways that people are trying to cure this. So I mentioned oral immunotherapy. Yeah. It's becoming more popular, where you're trying to basically just make yourself more tolerant by increasing both the amount and timing of that allergen. And I think there are other ways people are looking at this. I mean, like you can take a monoclonal antibody and you can help to block the ability of that IgE to bind to those Fc epsilon receptors, so that there's no release degranulation. There's a lot of different ways. I think then the most popular would be the EpiPen for very severe.
That's the immediate, right?
Yeah,
so
immediate, right.
You mentioned both immediate therapy interventions as well as long-term therapies. So this oral tolerance development in immunotherapy is more of a long-term thing. But if you're having a reaction right then and there, you need to get the histamines on board. You need to get the epinephrine on board. And as far as I understand, and it's a little bit out of my, I didn't go to medical school, but I think the idea there is that you have such vasodilation that the blood pressure drops. And so you're trying to keep the heart beating and keep the blood flowing so that the person doesn't collapse and go into shock. So I think that's the point of the epinephrine. And then you also want to get immunosuppressants on board to kind of shut down the immune response.
Right.
So I think those are the three main arms of the immediate therapy.
So then there's treatment, obviously, and then there's prevention. And, I think there's been a lot of debate as to whether if you are, well, I'll say this. Let's say that if you're a mother and if you're pregnant and what you eat, does that impact potentially the allergies that your children have? Because if we're talking about sensitization at an early age, which by the way, and you guys can tell me, I'm pretty sure back maybe 20 years ago, they said, don't let kids eat peanuts when they're young.
Right.
And now, of course, with what we know with oral immunotherapy, let kids eat peanuts and hopefully that will sensitize them.
Well, when I was young, they let me eat anything. Nobody worried about anything.
No one worried.
Maybe that's the key. Yes.
Maybe.
Ignorance is bliss.
I know my daughter, when she was young, had a lot of allergies. So we went to someone, I don't remember what kind of doc, but he had this apparatus to test allergies against, grass, wheat, everything. And it's a rectangular object pressed on her skin and
had little needles.
And then you have like an array of 12 little spots and then within, an hour,
her arm blew up.
Wherever she was allergic to something. Yep. And I remember watching and I was explaining to her what was happening. And the doctor goes, how do you know this?
All skeptical.
Come on, really?
Right.
And so then based on that, we could, and they tried sensitizing her by injections of the antigens, right?
Yes, you can do it that way as well. Yeah.
But I don't know if that would deal with oral intolerance, right? But anyway, in the end, she grew out of all her allergies, not allergic to anything anymore.
We don't know, right? So weird, right?
Yep.
Right.
But I think that idea of exposure, during either early during after birth or during development in the mom and what the mom is exposed to is really a fascinating area. And that's the subject of the paper that you wanted to talk about. We ended up talking about all of these ideas so much longer, but it's so exciting to talk about all these different aspects of immunology. Yeah. It's so complicated and to unpack all of these different things. And we really, in this amount of time we've been talking so far, have only just scratched the surface and identified some of the areas in where we really don't know a lot and some that we do. But yeah, I don't know if you want to talk a little bit about the paper now too.
Yeah, sure. No, that sounds great. So as we've been talking then, okay, could potentially what you eat during pregnancy affect it at all? And there have been some studies that shown that yes, in fact, in eating peanuts, your child may be less likely if you're breastfeeding to have an allergy. But then a systematic review came out recently, and it showed really that there's compliance issues with mothers following up, whether how long they're going to breastfeed, if they're not going to breastfeed. So it's really been a question as to if that really is true. And so what this paper sets up, their hypothesis, is that maternal allergen-specific immune responses influence food allergy responses in the offspring. Okay, so the title is Maternal IgG Immune Complexes Induce Food Allergen-Specific Tolerance in Offspring. The authors, I think there's just one first author, correct? There's two corresponding authors. One first author, Asa Osaki is the first author, and then the two corresponding, Richard Bloomberg and Michiko Oyoshi, and they are from Boston Children's and Harvard Medical School and Brigham and Women's Hospital in Boston.
And this paper is in Journal of Experimental Medicine this year, 2018.
Yes. Yep. Great. So I'll roll right into it. So what they did is they used mice as their model, and they took firstly 6 to 8 week old inbred lab mice, and they epicutaneously sensitized them with ovalbumin. And what that is, it's an egg protein. So they refer to it as OVA, and essentially trying to model egg protein allergy in these mice. As we talked about before, you can basically sensitize through the skin. And that goes back to thinking about this common mucosal immune system, and that the antigens that your immune system sees at any other site, you're going to have a response in other parts of the mucosa. So the first thing they did, because they to episensitize them, they basically took, I've not done this, but they took adhesive sides of tape, and they remove the top layer of the corneum. So that's the first thing.
Stripping your Band-Aid off of your arm.
Yes. Yep. And the goal is really, I mean, you're just trying to expose more of your skin so that more of the allergen can, because you have a lot of dead skin cells on top, and it makes a barrier for the dendritic cells and other antigen presenting cells to see the antigen. So they rip that off. And then
they
what
shaved
they did,
the mice first too.
They shaved it. Yeah, that's true. That would be mean if they were just ripping fur off these mice. And after they then, they shaved, they did the adhesive tape, they added the ova or saline to a patch of sterile gauze, and they added it to that site. And they did that every 3 days, 9 days before mating, and then once weekly during pregnancy and throughout lactation. So they did this in the pregnant mothers throughout that time. Then what they did is they took the offspring and they sensitized them as well as they as they were essentially growing. And then they challenged them at day 9. And they did that by gavage with either saline or, again, the ovalbumin. And so the question was, does sensitization with either ova or saline of the mothers impact sensitization or not of the neonates, and what happens when you challenge them? And so what they found was that sensitization of the offspring with ova from non-sensitized mothers resulted in food allergy. And they measured that by increases in all the things that we talked about before. So that would be ova-specific IgE, that Th2 cytokine IL-4. They measure that anaphylaxis, as Cindy said, by a drop in body core temperature, and some other indicators that looked at mast cells, both the numbers and the percentage, and then IL-13. So all of these parameters led them to say that when you challenge babies that were born to non-sensitized mothers, that they develop food allergy. But this was not seen in the babies that were born to sensitized mothers. And so what was responsible, they wanted to then move on and say, well, we know that this is happening, so what's the cell type? Now think back to, or go ahead back and listen to the previous segment where we talk about all these different cell types. And they wanted to know, is the proliferation in numbers of these T regulatory cells correspond to the ability of the pups born to sensitized mothers to not have this food allergy. And so what they did, if people are curious about how we in immunology work with these sort of things, so they took cells from the mesenteric lymph node and they stained them with a proliferation dye. And then they either add antigen, the ova, or saline. And they even used peanut allergen. I think they were trying to model another relevant allergen, which would be peanuts. And they add them together. And then essentially they wait for them to proliferate. So the cells that are going to be more responsive to the antigen, that are less sensitized, are going to proliferate more. And since you dyed them with a dye, there's going to be less dye because the cells are proliferating more. And so that's a way that you can, in culture, see how these cells are going to react and use methods to be able to determine that. And so what they found was then the animals that had less reaction had more Tregs, just as we talked about before, this being a really important cell type in downregulating those Th2 responses.
But does that, just because they're there, does that mean they're doing anything? Does that mean that they're really responsible for the phenotype?
Well, and so what they did, right. So what they did to move on from that, after they did a couple other functional assays, but they said, okay, well, we're going to delete these cells from the mice. And they did that by using a specific mouse where I had mentioned previously that there's a transcription factor in regulatory Tregs called FOXP3. And so what they did where they expressed diphtheria toxin receptor under the control of that FOXP3 promoter, so that in these mice, when you give them an injection of diphtheria toxin, it results in the elimination of almost all of those Tregs. It's kind of a cool way to delete those cells.
And the most important part about that is that mice, believe it or not, don't respond to diphtheria toxin. You can inject them with huge amounts of diphtheria toxin. It will do nothing to them. But then if you put the human diphtheria toxin receptor into the mice, now all of a sudden they're exquisitely sensitive to diphtheria toxin. So if you express it specifically in a particular cell that you want to delete, you can inject the mice with low levels of diphtheria toxin and all the other cells are perfectly fine, but only those cells that now express human diphtheria toxin receptor are deleted.
Right. And so just as they did before, same setup, and they challenged unsensitized pups with ova. And it was those animals that had an increase in susceptibility to the food allergen. So it essentially suggested further that the Tregs are really responsible for the decreased response. So then moving on, they asked the question, if the ingestion of the milk from ova-sensitized mothers resulted in the dampening of that Th2 Ig response, and it's due to the expanded Tregs, what is it in milk that is helping to expand the Tregs in the gut? So there has to be something in the milk that is then helping to expand Tregs. They bring up the concept of these immunoglobulin allergen complexes. So when you think about milk, there's a lot of stuff going on. There are immune cells, there are antimicrobial peptides, but there are also allergens, there's pieces of bacteria and viruses, and there's a huge content of immunoglobulins. So the allergen is bound to IgG in this immune complex in milk. And they do a series of experiments, in cross-fostering the pups to try to show that these Ig allergen complexes are what's then being transported to the gut to then stimulate the proliferation of Tregs. So by cross-fostering them, they immediately after birth, they took a portion of the litter pups born to unsensitized mothers, and they allowed them to suckle on ova-sensitized mothers, along with the ova-sensitized mothers' own pups. And the remaining offspring from the unsensitized mothers were kept with them. So they found then that the pups born to the litters of unsensitized mothers suckling unsensitized ingested ova-specific IgG1 and IgG2 complexes with allergen to the same levels as the ones born to the sensitized. So at the end, both groups, the cross-fostered and the ones actually born to the oversensitized moms, they had the same levels of these complexes. And they also then had the same levels of Treg populations in the lymph node. And so once they challenged the pups, they equally were protected, which showed that it was what was in the milk that was contributing to this.
So that was a beautiful description of that. But I think that you can probably say it one more time, because otherwise I think our listeners are going to have to rewind.
Sure. Yeah.
Do you know what I'm saying? I mean, it was beautifully explained, but even as complex as it is, I had to think about it a long time before I quite got all of the concepts there.
Sure. So basically the whole goal was to take babies born to a mother that had been unsensitized, throw her on the teats. Do they call them, in pigs we call them teats. What would they call them in mice? I don't even know.
Not sure. Nipples? I don't know.
Nipples? I don't know. But they would basically have them suckle the ova-sensitized mothers. So if you have on one mother suckling babies born from her, she was sensitized, her babies were born sensitized, and then you put on her babies that were born from unsensitized mothers, and they both end up having the same amount of these immunoglobulin allergen complexes that come from the milk in their intestines. They both increase the amount of Tregs inside their gut, and they both are protected when they are challenged with the allergen, which is the ova allergen. What that demonstrates is that it is what these complexes in the milk that are helping to protect both pups, or essentially from the ova challenge.
And in fact, they can't find any free ova in the milk.
Right.
It's always a complex.
Right.
They actually looked for it, right?
That's right. Yeah. They did. Mm-hmm.
Right. Now what's interesting, and I'm going to go through I don't know how we doing on time. You can tell me to speed up.
Long, but it's okay.
It's fine.
Okay. Well, essentially, I may skip through this part, but I will talk about, there's not only passive transfer of these immunoglobulins happening in milk, it also happens in utero. And so what they did a series of cross-fostering experiments where really what they found was that it's not all to do with the milk, that there actually is some passive transfer of these complexes that happen transplacentally, and that helps to protect the pups intermediately. But really, it's the milk that helps to optimally protect them. So I won't go through all those experiments, but it just goes to show that to optimally protect them, they needed both transplacentally and the milk IgG complexes.
But I guess therapeutically, the important thing is you could transfer it in milk, suggesting that if you supplemented, if there's a lot of bottle-fed babies or whatever, if you supplemented them with something like these immune complexes in the milk that they're drinking, that might protect them, even if they didn't have that exposure in utero or were not breastfeeding.
Yep. And they actually did that. They made their own immune complexes. They took immunoglobulins, complexed them with allergen, and they gave them to the moms when it either... Well, they didn't. There was a third treatment group they should have done, but what they did, they gave it to them either throughout pregnancy and lactation or just in lactation. And they showed that by adding that, it helps protect the neonates. Now, of course, I would have liked them to see just do it during pregnancy because not lactation, because that will really further define the transplacental. But I don't know if they just wanted to let's just not talk about transplacental because it doesn't really help us with the milk story. So it would have been, I think that's a future research project that maybe would be interesting to do, is how placental transfer works.
And remind me, in your pigs, do they do placental transfer? Because some animals do and some animals don't. For example, I know in horses, there is no transplacental transfer. And so if the foals aren't able to milk, they're extraordinarily sensitive to various different diseases after birth.
Absolutely. So in swine, as in bovine, the placenta is like 6 layers thick. And so there is no immunoglobulin transfer in the placenta. So any passive transfer happens postpartum.
Wow.
Yeah. So it's amazing. They get all they need from that milk. And so if you basically take a calf or a piglet and you don't let it suckle, it does very poorly. There's just no development in the intestine. So depending on what species you use, it can be very interesting in terms of differences.
So it's because the placenta is so thick in pigs, does this prevent, say, viruses transferring from mother to offspring?
Now, viruses are just if we could anthropomorphize, they are very smart and tricky. And there are definitely, so there's this really problematic virus. It's an arteriovirus called PRRS. Yeah. And PRRS is able to cross the placenta and cause spontaneous abortions. So there are viruses that have been able to work their way through those 6 layers and be able to cause problems. So
I have a question about this idea of oral therapy, right? So this experiment, they supplemented complexes, right, to the mothers. So the thing is, here they're working with ova, but what would you do if you wanted to treat people? Which complex? Would you give them a cocktail of everything that is known to be problematic, or what? Because your baby's not born yet, so what would you do?
I don't know.
You mean in terms of what would you give the mothers?
If you wanted to use this as therapy in people down the road, right? Would it be specific, or would it be a cocktail of antigens, do you think?
I guess you'd approach it like a vaccine, right? So whatever the most dominant antigens are that would be protective, and I don't think we know those yet, but you would use those. Just like when you're developing a vaccine, you try and choose the immunodominant antigens or epitopes that drive the most protective immune responses in the vaccine. So I don't know what they would be, but I could imagine if a lot of the details of this are worked out, one could identify the most active and protective epitopes and make a cocktail of those. And now they'd probably need to be complexed with monoclonal antibodies that were produced. But then you could take those immune complexes and deliver them via, milk or bottle feeding or whatever. But we're not there yet.
So in this experiment, they injected the mother's IP, but you wouldn't do that, obviously.
Right.
Would you actually would mothers drink it, or would you feed it to the children, do you think?
I think you could probably feed it to the children because you wouldn't necessarily need the mom. Yeah. This is being delivered from the mom, but you could gavage it as well.
Okay.
Right. So just to kind of wrap up this paper, they talk about some of the mechanisms of how this could be happening. I had mentioned before that FcRn receptor, or neonatal, it's called the whole term, it's FC, it's neonatal crystallizable fragment receptor. And it helps the bidirectional transport of IgG. And so without that, they knocked that out in mice. The pups could swallow the milk, but the IgG could not be uptake into the lumen, to the lamina propria. And so that helped to show that receptor was important. And what was interesting, and something I did not realize, was that dendritic cells actually express FcRn.
Yeah, I didn't realize that either.
I had no idea. I really just thought it was I mean, there's a lot of different cell types, but I thought they were really limited to, barriers like placental barriers or intestinal barriers. And so what they did was they were able to knock them out in dendritic cells and show that the dendritic cells, they can't even uptake when you knock out that FcRn, dendritic cells cannot uptake and process that. And so it's really important in the transporting of these complexes through the dendritic cells and then onto that MHC complex. And so they identify a particular dendritic cell. It's called CD11C DCs, which I had mentioned before, that these are really the specific type that are important in expressing FcRn to help bring that complex in, process it, and present it. So they did a little experiment at the end where they are looking at, I'm sure the reviewers were asking about,
oh, yeah.
So
how does it have to do with humans, right?
How does this have to do with humans? And I will say one point in this paper, if you guys read this, they did not address this at all, but I think it really is important. So in mice, as well as in pigs greatly, but in mice, the guts of those animals don't close until weaning. So in baby
so what do you mean
humans,
by close?
Okay, yes. So basically the tight junctions don't form to the level of a mature mouse until they wean. So essentially, I'm assuming there's some growth factors in milk that help the maturation of that process. And so the transport of immunoglobulin or immunoglobulin complexes actually is the rate of transfer is higher than what we would probably see in a human. They really see gut closure. And if you could see me, I'm doing those little quotes, up to it's like 3 days of age. If I have that correct, it's like 3 days. And there's really not a lot of transport of Ig after that. So in pigs, it's up until 24 hours when the colostrum is being produced, that the guts are just, the tight junctions have not formed, they've not closed all the way. And the biological purpose is those animals did not get any immunoglobulin transplacentally, so the guts have to be really open. And they never really brought this up, that if human babies likely cannot uptake these complexes as efficiently as what they saw in these mice. And so while this is really interesting, I think that this also is a function of species differences, and that we would need to create, and we do this, humanized mice to more of a degree. They did use humanized mice. They basically put the human receptor for FcRn so that they could better model a human response, but they would have to really alter the gut, in my opinion, to help it close faster to see if this is really true in humans.
Yeah.
That was the biggest thing I had to say is it's really interesting. I felt that the experiments were well done, except for that biological difference in mice and humans is complicating.
So that in that experiment, they took human milk.
They did.
Yeah.
They showed OVA Ig complexes in them, right?
Yep.
Yep.
So presumably those mothers had been eating eggs, right?
Yeah.
And then you could give them to these mice that you described, and it protects them against allergic responses, right?
That's right. And if you took the IgG, if you took the human milk, and you took all the antibodies out and then gave it to the mice, they were not protected. Right. So it was definitely the human mothers who had consumed eggs made these immune complexes that they could then transfer via the milk to a baby, theoretically, if their barrier was quote unquote open, right? Yeah. And so at least in the mice, they could transfer that, and it was dependent on the antibodies.
Yeah. So Steph, you're saying that human babies, this wouldn't work because the gut is already closed, or would it work less well?
And this is why I really wanted them to see, more experiments where they were really focusing on the placental transfer, because I think, you can get around that potentially by targeting certain gestational time points where the rate of transfer in the placental is increased. And that could help get around the fact, breast milk likely does contribute to allergen tolerance, but I just don't know as to what effect, as what they've seen in this paper.
Right.
Well, that's important, because if you want to do this in humans, you're going to have to, as we have said, feeding the babies would be easiest, but if you have to figure out how efficient it's going to be. Maybe you would not be able to do that. Depending on the outcome, you'd have to go back to mothers and think about feeding them. Yeah.
That's right.
So If people are looking for an executive summary of this paper, what they basically indicate is that allergen-specific IgG, immunoglobulin complexes with allergen in breast milk help drive this allergen-specific Treg response, and it's food-specific IgG antibodies that exert basically this induction of tolerance against food allergy. And it supports the concept that FcRn receptor on the surface of dendritic cells are helping to make that happen in bringing in those complexes so they can be presented to the Tregs to help proliferate and keep that Th2 allergen response down.
So I think somewhere in this paper they say, in the discussion, our results provide experimental support for recent decisions to withdraw recommendations of allergen avoidance during pregnancy and breastfeeding. Right.
Right.
So that brings up my question. What if the mother has allergies herself, right?
Right. Yeah. Well, then she can't
do it then,
she
right?
can't consume the.
What's interesting is that, and this has been actually told to me by a couple friends of mine who are pregnant right now, they were allergic to things like cow's milk, things like grasses. And when they are pregnant, they are no longer allergic. And it could be due to the fact that during pregnancy, your immune system changes quite drastically. You actually,
absolutely.
Yeah, it's defined by an increase in hormones like estrogen and progesterone, which help to increase Th2 responses, regulatory responses, and it can dampen the effect. So, it's definitely need more research because it's interesting.
This is a great paper. I really enjoyed it, and
good,
clearly written. And I like that every experiment they start with a hypothesis. They set up the experiment, just like we try and teach students to do, but don't often. This is the hypothesis. Here's the experimental design. Here are the results, and then they move on. So
and their experiments were so incredibly complex at some point.
Huge.
They just had these beautiful figures describing the setup of the experiment. Because when you're cross-fostering moms from sensitized and unsensitized onto sensitized and unsensitized, and then it was incredible, the complexity of the types of experiments they designed, and they were so well designed to answer their questions. It was really nice.
Absolutely. Yep. That was my favorite part, was the amount of controls. And I felt like at the end that we got the answers we got to answer the hypothesis. So great paper. I'm glad you guys liked it. I think it has some good implications and also reflects some of the things that I'm interested in. So I appreciate it.
Well, that's the best thing to talk about, what you're interested in, right?
Yeah.
That way you can speak with some authority. as you did. Thank you very much. That was great.
You're welcome.
Yeah,
sure.
thank you.
Yeah, absolutely.
We have a little time. We could read a couple of emails. First, we have a few follow-ups from our CAR-T episode. The first is from Bob. Kudos on this episode on CAR-T immunotherapy. It was wonderfully detailed and absorbing. At one point, the discussion touched on regulatory T cells and the autoimmune response. I'm interested in this area of immunology since my daughter has late-onset type 1 diabetes thought to be an autoimmune disease. A recent article in Scientific American, February 2018, speculates that an enterovirus infection similar to polio is involved in triggering the response. Here we have a perfect confluence of topics dear to podcast members, viruses and the immune response. Might it merit a show?
Oh, I think that would be interesting.
Absolutely.
We could possibly do that. I will look into that, Bob. That would be a natural for me to do, I guess, right?
Yeah.
Cindy, can you take the next one, please?
Sure. Hi, immunomodulators. I love that. That's great.
That's great.
So this is from Niraj, and it says, I hope your 2018 is going great so far, and I was absolutely thrilled to listen to the most recent immune podcast on CAR-T. This is truly a remarkable breakthrough, and I am glad that you had such a nicely outlined and detailed discussion on the same. Cancer plagues humanity at so many different levels, and to have a breakthrough like this actually transform into curable therapies is totally miraculous, both at a molecular and emotional level. I realize that this technology is still in its infancy, but I am certain that with time, alterations will happen that will make it safer, more efficacious, and more importantly, affordable. The current price tags, whether it's for the Novartis or Gilead's therapies, are obviously exorbitant. But given the immense technological challenges one overcomes in performing them, are probably a bit justified. At times, it's easy to demonize the drug companies. And yes, EpiPen price escalation was hideous. But currently working at one, I can vouch that this is an incredible capital-intensive enterprise. When things succeed, it looks great. But one never sees the countless dollars and man-hours that went into overcoming challenges that don't succeed. Yes, we learn from our failures, but when the investors and stakeholders are breathing down your neck, it isn't always memorable when your R&D efforts fail to justify the research undertakings. I think that detail is sometimes amiss to the broader audience, and just seeing an exorbitant price tag for an approved therapy can at times certainly influence mindset. But CAR-T, even at this early stage, feels like a unique opportunity that is moving ahead at breakneck speed. And I hope for patients that this is just the start, with many more improvements in the near to long term. And in light of talking about progress toward treating cancer, I am in agreement with Dr. Racaniello about his recommendation for the book Emperor of All Maladies. This is probably one of those books that everyone wanting to learn or having impacted by cancer should absolutely have on their reading list. In my opinion, it is one of the best orations of the history of a field of scientific investigation as diverse and ancient as cancer. For me, it is second only to the history of how genes were discovered and how understanding the circadian rhythm as listed in Time, Love, and Memory by Jonathan Weiner. Thus, in closing, I would like to personally thank you for taking the time from your busy schedules to release these podcasts. It doesn't include you, Steph. As being a graduate student, your time right now has no intrinsic value except when the experiments succeed.
Thanks, Raj, for the potentially heartbreaking realization. Yes.
I guess that's pretty much how most graduate students feel. Overall, the entire TWE Immune X series is something that every curious soul should listen to, as I am certain that it will benefit you in ways you can't even fathom. Thus, keep up the great work, and I hope you have more sponsors for these pearls of wisdom. Best, Niraj. P.S. And maybe just a suggestion, someday you can have a discussion on flu and why it is so hard to generate highly efficacious vaccines to induce long-term immune memory against the seasonal epidemic. It is probably not as tricky as dengue, but nonetheless, it overcomes our immune surveillance quite effectively if you are not vaccinated or even at times when you are. I think that's a great idea because, combining those influenza as well as the immune system, if we talk about that with expertise on both sides, that would be really fantastic, I think.
Yeah.
Okay.
Vincent, are you influenza'd out?
No, not at all. I think it's very interesting, and having done my PhD on it, I could never be influenza'd out, right? my first viral love. Steph, can you take the next one?
Yeah, sure. How do you think you pronounce this name?
Yesel?
Yesel. Hello, Vincent, Cindy, and Steph. I cannot thank you enough for creating the Immune Podcast. While I listen to all Twix shows, immunology is something I have always been fascinated by. I'm not a scientist yet, but an oncology nurse, and so the content is more familiar and relevant to me. I'm especially thankful for the CAR-T episode. I've been closely following recent developments in immunotherapy over the past few years, and CAR-T cell therapy is by far the most mind-blowing. I especially appreciate your efforts in presenting the material in an everyday language without dumbing things down. Agreed. Cindy did great. Some of the classes I teach at our hospital include immune system, stem cell transplant, and chemo drugs. And I'm learning a lot from you, not only the content, but also how to present it effectively for non-scientists. Because I enjoy this episode so much, I have quite a few comments. First of all, there's a question as to why chimera, is it chimera? Chimera, thank you. Chimera is indicated specifically for children and young adults. Mm-hmm. It seems to me that it just has to do with the fact that it's approved for acute lymphoblastic leukemia, or ALL, which does primarily affect young people. As Cindy described, ALL is often curable, but there is a small subset that is difficult to treat, and treatment options are limited for relapse and refractory diseases. There are 4 main types of leukemia, and they have different epidemiology and characteristics, which I will summarize below. Is this something we could put in the show link, or should I go through?
Yeah, we'll put it in the show link so you don't have to read it. Just say the 4 types. That's all.
Sure. Acute
Myelogenous.
Myelogenous, thank you, leukemia, chronic myelogenous leukemia, acute lymphoblastic leukemia, and chronic lymphocytic leukemia. Thank you, CLL. Yescarta by Kite, on the other hand, is indicated for diffuse large B-cell lymphoma, which occurs in adults. I'm curious, though, why two different agents for two different diseases? If both agents can recognize CD19, wouldn't that mean that they could be used for cancers of CD19 positive cells?
I think that might be a patent thing.
Yes, I would agree. I'd also like to make a comment about implications of all these new immunotherapy agents like checkpoint inhibitors and CAR T cells, because their side effect profile is drastically different from what people are generally familiar with from cytotoxic chemotherapy. Educating the public, not only the patients, but also healthcare professionals beyond oncology, is critical. Patients must understand how important it is to report even benign symptoms, maybe a little bit of skin rash or diarrhea, which may seem like nothing at first, but can be the first signs of something more serious, a more serious inflammatory immune response. I've also heard of fatal cases where non-oncology physicians failed to recognize the signs of inflammation, due to checkpoint inhibitors, and it was too late by the time the patient's oncologists were contacted. That's very sad.
I think I tried to mention that, but I probably didn't emphasize that enough. But yeah, what oncologists are used to in traditional chemotherapy and all of the side effects that they deal with that are completely different. So it is an entirely new world when you're dealing with these patients that you're putting on these types of therapy, whether it be a checkpoint inhibitor or a CAR T cell. When you're taking the brakes off the immune system, it's just completely different. And so in one case, you're looking at immunosuppression and worry about exposure to infections. In the other case, it's really releasing the brakes on the immune system and allowing some autoimmune inflammatory responses to occur. And so understanding that difference and being able to treat them, which is completely different approaches to treating them, is really important. And so there's a whole discipline of oncologists that are developing who are specializing in treating patients that are on these immunotherapies.
Well, there's some more questions for you. By the way, a question for Cindy. Have you encountered a drug called blinatumomab during your research for this episode? In my mind, I thought of this drug, which we started seeing last year, as a precursor to CAR T cell therapy. It's called bispecific T cell engager, or BiTE, and has CD19 and CD3 binding sites. Would you say that this is the same thing as the bispecific antibodies discussed in episode 3? Also, blinatumomab is a slow, continuous infusion for 28 straight days. Patients go home, that's a lot, with a portable infusion pump, which isn't very convenient. Any ideas why this is necessary?
So I didn't have a chance to look this up because I didn't look at the emails before we started. So no, I had not heard of blinatumomab. But I had heard of BiTE, the bispecific T engager, and the idea that you're hitting two things, CD19 and CD3. You're hitting both a T cell and a B cell. So CD3 is expressed on T cells, CD19 is expressed on B cells, and so yes, I would say this is probably a bispecific antibody similar to the Zika bispecific antibody we talked about previously. I don't really know why it's a slow continuous infusion. It may simply have to do with whether you are continuously treating and trying to keep those T and B cells down, or whether you allow them to re-expand and then decrease their numbers again, and allow them to re-expand and decrease their numbers. Because remember, as you mentioned when we started, there's a reservoir of hematopoietic stem cells in the bone marrow that can repopulate the immune system. And so they're continuously generating B cells during life. And some T cells, although the majority of T cell development happens during development as a fetus. So I don't know the 28-day continuous infusion. I can't give you an answer for that. I'm sorry.
And then one last comment. I ran into this paper, and it's linked here some time ago, which explores combination of PD-1 checkpoint inhibition using CRISPR-Cas9 and CAR T cells. It's a straightforward experiment. Well, speaking from a person who has no idea what entails to conduct wet bench experiments and logical next steps of sorts. On the other hand, it seems like a scary idea to me to permanently disable the checkpoint function of T cells that are being released out there to kill stuff, especially if we don't know that the transferred CAR T cells still circulate in the body even after 10 years. I'd love to hear your thoughts on this as to the pros and cons, whether this should be done or whether this should become reality clinically. Thank you again for everything you do. You're a fan from Los Angeles, Yesel. And P.S., would you consider an episode on graft versus host disease? Absolutely.
Definitely.
Yeah, it really relates to T regulatory cells as well. Coming from a bone marrow transplant nursing background, GVHD is the one thing I both love for its mechanism and hate for what it does to people. I have had patients whose skin literally sloughed off, which is acute GVHD. To those who die slow, agonizing death from bronchiolytic obliterans or chronic GVHD. Emotional aspects aside, I would love to know what exactly the immune cells are doing to bring these effects. Oof, that's awful. Wow.
So Cindy, what about this PD-1 CRISPR experiment? Do you know anything about that?
I'm not sure about that.
Well, what they did was took T cells and besides, they just CRISPR'd out PD-1.
Yeah.
As a way of getting better killing, right? With CAR T.
So
at the same time,
I don't know if this was the case, and I'd have to go look at the details of that. But I would imagine it comes back to when we talked about those kill switches. So if you do that to those CAR T cells and you take off the PD-1 and you put those back in the body, I think you have an ability to get rid of them with a drug in case things go wrong.
Right.
So I think it's rare now that we put these things and unleash them in the body without the ability to just eliminate them quickly should they start to cause a problem.
And I've seen, and these experiments were done in mice, and I've seen a lot of my non-scientific Facebook friends posting. There was recently this big blurb about CAR T cell therapy in mice and how, in addition, I think maybe to checkpoint inhibition, they were able to clear all tumors 100% effectively. And so I commented on a couple people's, I said, well, this is done in mice. And actually, a lot of the previous immunotherapies, like IL-2, giving IL-2 to try to combat the tumor in mice worked, but given to humans was a really bad idea. So I think we have to be able to do this in the clinic. And of course, it's like you said, a new territory. So we have to proceed with caution with our interpretation.
And I think that comes back to our comment from Niraj, right, who worked at the company where, things that work incredibly well in basic research in mice, they give us hope. There are all these great ideas that come out. And as we start to develop those therapies more in a biotech company or a pharmaceutical company, we may start and often do realize that they're not going to translate. And so when you think about, somebody describes it as like a cliff. So you come up on one side with the basic science and then bridging that gap and making it into the transition to becoming a therapy is an incredibly large jump with a deep valley. And many of the therapies end up not making it over when they try to jump. And so if 1 out of 100 or maybe even fewer than that actually make it, that's where the costs come, right? Because you can't fail to try and develop something that looks so promising, but then if you get down the road and it doesn't work and you've invested in a lot of money in it, we've learned a lot about how the immune system works in human and all of that. And it's incredibly informative to the people who are in this knowledge gathering aspect of science. But for those that are required to make these therapies happen, it's failure after failure. And then you get one gangbuster, blockbuster drug and you have to try and financially manage the R&D versus the actual clinical transfer.
Right. And a little side note for Yesel, I just want to say, I really appreciate what you do as a nurse. I mean, I can only imagine the emotional toll that would take to have to see these patients. So make sure you do a little self-care yourself so that you're able to care for those patients. So great job.
Next one's from Mark. Episode 4 was amazing. The presentation on CAR T cells was outstandingly well presented, and the subject matter blew my mind. After the episode, I spent hours pondering in amazement. Great. That's what we want to do. Could I ask, is it correct that T cells initiate cell death in a different way than occurs during apoptosis? In apoptosis, there's a managed deconstruction process which occurs within the cell, right? I'm guessing that T cells employ more of a demolition process than a deconstruction process. To use an analogy, blowing up the cellular building with explosives rather than taking it down floor by floor with power tools, the resulting debris setting off the immune system.
Can I take that?
Yeah, go.
So believe it or not, we don't want to destroy the building because when the contents that are inside of a cell get outside of a cell, it alerts the tissue that something has been damaged, and it needs to induce a repair response, which engages the inflammation component of the immune host defense. And so T cells do, in fact, use apoptosis to kill their target cells. And so what happens is we talked about the T cells being able to secrete factors that poke holes into the membrane of the target cell and deliver the death signal. And that death signal is something called a granzyme. And that granzyme activates the caspase cell death mechanism within the T cell. And so it doesn't just explode, but it is a deconstruction, and it's a more quiet and controlled cell death. That being said, that's not the only mechanism. There are things called pyroptosis, and it's called pyroptosis because it causes fever. So when you have this lysis of cells, it can induce this inflammation and systemic inflammation. But the large majority of the time, a T cell, for example, will try and kill a virally infected cell in a quiet and controlled way.
Okay. And one more from Mark. Another thing, when you guys talk about allergies, could I ask a preemptive question that you might be able to work in that discussion? I receive allergy therapy in the form of a monthly set of injections that takes around 5 years. It's for seasonal allergy to pollen cats and a dust allergy. If I already have memory B cells generating the wrong response to allergens, how does this treatment work to reduce the population of those B cells? Maybe someone can develop CAR T cells to target the BCR of cells which match a particular antigen to quick reset the B cell population instead of 5 years on injections.
Right. Well, I mean, that you do have memory B cells that potentially are generating IgE, which is releasing the degranulation of those mast cells. And really, as we talked about before, because you're having increasing amounts, he said monthly set of injections over the course of 5 years. The goal is to basically develop an increased amount of those T regulatory cells and continuously prime those dendritic cells over the course of time so that they become desensitized to that allergen. So hopefully what we talked about today fits into your question. Sorry, we didn't read that beforehand, but I think in the episode we touched on it.
Okay. Keep up the amazing job. This is my favorite of Vincent's podcast, and I've listened to them all simply because the subject matter of the immune system is so awesome. Mark is in New Zealand.
I totally agree.
Yes, the best.
Steph, do you want to take Anna's?
Yeah, I'll take Anna's. Dear Cindy, Steph, and Vincent, thank you for starting this podcast. Immunology is one of those simultaneously fascinating and daunting subjects for me. Don't worry, Anna, that's for me too, and probably many others. I remember writing a short essay at university about dendritic cells and finding it so interesting, but also very complex, that it was hard to make sense of all those cells, molecules, and pathways. I'm afraid I'm still a complete ignorant of the subject many years later. I have a question. It has been repeatedly shown that breastfeeding is beneficial for babies, helping to protect them from pathogens and even stimulating the development of their immunity. However, working with measles as a virologist, I know that, for instance, if the MMR vaccine is given before the recommended 12 months in the context of an outbreak, it should be given again at the recommended age as the maternal antibodies present in the child. It should be given, let me read that. It should be given again at the recommended age as the maternal antibodies present in the child limit the effectiveness of that early vaccination. This made me wonder, how does passive immunity gain from maternal antibodies before birth and through breastfeeding work with or for or against the child's own active immunity? And this is a perfect question for this episode. That's and right. Of course, the benefits, the pros of being able to passively transfer antibodies, both transplacentally and through milk, is that they are going to bind complexes and remove them likely in the gut, or they're going to then go into circulation and help to remove and bind and neutralize pathogens in other places. What she's mentioning is true, though. If you have circulating antibodies to part of the measles virus, you are likely not going to develop as effective as immune response as if you were not, if you did not have those circulating antibodies that you got from your mother. And there's two things that do this. One's called epitope masking, and the other one is cross-linking. I've mentioned cross-linking earlier in the episode. There are some other postulated ways that this maternal antibody basically blocking the ability of the antigen in the vaccine that you give to be recognized by of the immune system because the epitopes are masked by antibodies binding them. The other way is that you can actually have maternal antibodies that bind to a B cell and cross-link its receptor so that it cannot see the antigen being presented to it. And it's this really interesting phenomenon when we are trying to protect infants from disease, because you have this problem where maternal antibodies can effectively block an effective antibody response from a vaccine, but you want those babies to be protected. And so some of the things that are done in the field is you either give a higher dose, or you are going to give a booster, or you're going to try to wait until, like she mentioned, about 12 months, because those antibodies start waning about 6 to 8 months, and they're effectively cleared by 12 months. But that's if you're not breastfeeding up to a year. You could have those longer if you're breastfeeding. So, yeah, there's definitely pros and cons. And when developing vaccines for infants, you have to keep that in mind. Okay, I know this does not relate to any of the previous podcasts. Well, luckily it does for this one. So feel free to ignore it or keep it later for a more relevant episode. Many thanks for getting this going. Anna, fully immunized.
Wonderful.
All right, good job.
All right. That is immune number 5. You can find it at microbe.tv/immune. If you want to send us questions or comments, immune@microbe.tv. Please subscribe on your favorite podcatcher on your phone or tablet or whatever you use. Subscribe to us so you get every episode. If you like what we do, you can support us financially. Go to microbe.tv/contribute. On Immune Today, Cindy Leifer at Cornell University. You can find her on Twitter, Cindy Leifer. Thanks, Cindy.
Thank you, and stay immunized and stay away from the flu. It's bad this year.
Steph Langel is at Ohio State University. You can find her on Twitter, Stephanie Langel, all one word. Thanks, Steph.
Thank you all. This is great.
And I'm Vincent Racaniello. You can find me at virology.ws, Prof VRR on Twitter. The music you hear on Immune is by Steve Neal. You can find his work at stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. See you next month.